Well, good morning, everybody. Hopefully, everybody's doing okay. So, today we're just going to continue right off where we left off last time. We're going to continue our discussion of nucleotides. And today we're going to kind of go into, a little bit more of the health related aspects of them and go over some overarching concepts here in the class. Um, Before we move forward, just to reiterate again, this upcoming Monday, we're going to be back in person. So we're going to be meeting in our usual room in Toldo at 8:30 a.m. on Mondays and Wednesdays. Class will be back in person. So everything will be back to normal. So like I said earlier or last week, when I come back, I'll put an Excel sheet up on Brightspace so that everybody can, you know, try to book a time that works best for them to do the exam viewing so you can look at it over um, and whatnot. But without further ado, let's get on with the lecture today and move forward. So I've decided that the next lecture, the next topic, 100% is going to be carbohydrate biosynthesis. Um, I was looking at the content, and lipid synthesis didn't really make too much sense right after nucleotide. I feel like the carbohydrate biosynthesis kind of just, is the next logical place to go after, we did our amino acid synthesis and now our nucleotide synthesis. So the next logical one to do, I think, would be carbohydrate synthesis. So we will start that next. We might touch about it today, depending on how much time, we get through. But if we don't, then on Monday morning, we'll, start bright and early with carbohydrate biosynthesis. Well, let's continue. Okay, let's start. Let's see if we can get this laser pointer going. Wonderful. Okay, so this is our enzyme that we've been talking about lately. This is our ribonucleotide reductase. So ribonucleotide reductase is notable in that its reaction mechanism provides the best characterized example of the involvement of free radicals in biological transformations. So before, um, like we said a little bit earlier, we thought that these were so rare in biological systems, we thought this doesn't happen. But understanding this ribonucleotide reductase, we noticed that there is involvement of these free radicals in biological transformations. So this enzyme's in E. coli and most eukaryotes as well. And it's usually in this alpha 2 beta 2 dimer, as you can see right here. So we have two alphas, so alpha 1, alpha 2, beta 1, beta 2, with catalytic subunits um, and radical generation subunits in alpha 2 and beta 2. Um, and this diagram right here, or this image right here, best represents that A2, B2, right? So the alpha 2, beta 2 dimer that exists right here. So each catalytic subunit contains two regulatory sites. Um, and the two active sites of the enzyme are formed at the interface between the catalytic, so the alpha 2, so this alpha section over here, and the radical generation beta subunits down here. So at each active site, an alpha subunit contributes two sulfhydryl groups required for activity, and the beta 2 units contribute a stable tyrosol radical. So you can see over here, we have this radical pathway over here, and then over here in the alpha part, um, we contribute the sulfhydryl groups up here. So over here we have the beta 2 subunits that have that binuclear iron, that Fe3 plus cofactor that helps generate and stabilize the tyrosine 122 radical that you can see right here. So in this beta 2 subunit right here, you can see the tyrosine 122. It's stabilized by that binuclear iron, Fe3 plus. And the reason for that is um, the tyrosine radical is too far from the active site to interact directly with the site, because if we remember correctly, the site of activity is actually in between, as you can see right here where my laser pointer is, between the alpha 2 and the beta 2 subunits. That's where the active site actually is. So having this tyrosine 122 over here is actually too far from our target. So we need a way to get there, right? And how do we do this? We do this through several aromatic residues, which form a long-range radical transfer pathway to the active site. So If we look closely in this figure right here, we can see the likely path of radical formation from the initial tyrosine 122, which is centered right here in the beta 2 subunit, um, to the active site, this cysteine right here, 439, which is used in the mechanism. Um, so several aromatic amino acid residues participate in long range radical transfer from the point of its formation at the tyrosine 122 residue all the way to the active site where the nucleotide substrate is bound. In previous slides, we talked about the regulatory role of this enzyme, ribonucleotide reductase. Um, and the regulatory effects that this enzyme has are accompanied by, and most often than not, mediated by large structural rearrangements in the enzyme, right? We have that, fundamental concept and, in molecular biology or biochemistry or even in science, right? That structure dictates function. So if the structure of something changes, it's more often than not that the function changes. And that's pretty much, um, an axiom of biochemistry, right? It's a law. It's pretty much accepted. It's never been questioned. It has been questioned, but it isn't, and it tries to be challenged, but time after time, again, it proves right that in order, that structure does dictate function. So when the active form of the enzyme, so this alpha 2, beta 2, is inhibited by the addition of, the allosteric inhibitor DATP, like we talked about before, a ring like alpha 4, beta 4 structure forms. So we end up from this, typical formation of how the enzyme normally would look over here to this ring structure we can see over here forming that alpha 4, beta 4 conformation. And you can see over here that you have these alternating alpha 2 and beta 2 subunits going all the way around this ring like structure. So, in this altered form, right, this altered structure, the radical forming path from alpha to beta um, is disrupted, right? And the residues in the path are exposed to solvent. So before you could see over here in this image that specific residues, whatever they may be, or specific sites on this enzyme or this protein, clearly, some of them are exposed to, that aqueous environments. You have those hydrophilic amino acids, right? But you have those hydrophobic amino acids and residues, on the inside, not exposed to solvent, right? But when it's disrupted, the residues in the path are now exposed to the solvent. Nothing is now protected from solvent anymore. Effectively, what this does, it prevents radical transfer and thus inhibiting this reaction from even moving forward. So the formation of a ring-like alpha 4 beta 4 structure, however, though, is reversed when DATP levels are reduced. So when DATP levels are high, we shift into this ring structure. This enzyme no longer works because we have exposed to aqueous environment. So the free radical transfer is inhibited. However, though, when DATP levels are low, um, this is reversed, as you can see through the reverse mechanism here, and we're actually able to gain functionality back in this enzyme that we once had lost before. Okay, so moving on a little bit here, shifting gears, right? We're going to focus now on the degradation of purines and pyrimidines, which produce uric acid and urea, respectively. So, purines would make uric acid, and pyrimidines would make urea. So, purine nucleotides are degraded by a pathway in which they lose their phosphate through the action of 5-nucleotide sidase. So, we have this first enzyme right here that occurs in both pathways. So you have 5-nucleotidase and you have 5' nucleotidase over here. And each one is the starting path for losing phosphate for either purines or pyrimidines at the very beginning. So over here, you can see that if you start off with AMP, so adenylate, which ends up yielding adenosine. So you can see right here, you have the removal of water. So you end up with adenosine over here, which then from over here gets deaminated to inosine by adenosine deaminase. And inosine over here is hydrolyzed to hypoxyxanthine. Hopefully, I'm pronouncing that right. So yeah, hypoxyxanthine. Um, so yeah, hypoxyxanthine and to ribose. So it's very key over here that we consider that we're also forming D-ribose and hypoxyxanthine from this reaction. Um, From there, hypoxyxanthine is then oxidized um, successfully to xanthine over here, specifically the keto form. And then from here, uric acid eventually comes. So if you follow the pathway here, it starts here where you follow my laser pointer, comes here, goes to xanthine, and then we should go over here. Actually, I think I can draw this out. So we go from xanthine, perfect, and then we go to here. Um, to this step over here where we use xanthine oxidase, which is a flavoenzyme with an atom of molybdenum and four iron sulfur centers in its prosthetic group. From there, molecular oxygen is the electron acceptor in this complex reaction. So you can see right here we have the addition of water, molecular oxygen. We've released this peroxide here, and we end up with the final molecule, uric acid from AMP. Now, for GMP over here, um, so just like before, we start off with GMP over here. So GMP catabolism yields uric acid as well, right? So if we follow this pathway over here, it's the same thing. So let me draw this out, maybe in a different color though. So let's choose this purple right here. So GMP would follow here. It would go here. It would follow this pathway, eventually come here, and then end up over here. Okay. So yeah, like I said, GMP catabolism also yields finally uric acid as the end product. So GMP is first hydrolyzed to guanosine right here. So we have that introduction of water, that release of the PI. Again, make note of that enzyme right there, that 5' nucleotidase, which plays a big role there. Guanine then undergoes hydrolytic removal of its amino group to yield xanthine. So it goes from this guanine product over here, and it ends up to xanthine right here by that hydrolytic removal of its amino group, which then again ends up being converted to uric acid by the xanthine oxidase over here. And then it's the same thing as before, where that xanthine oxidase is a flavoenzyme, and it has an atom of molybdenum and a 4 iron sulfur center in its prosthetic group. And again, molecular oxygen is the electron acceptor in this complex reaction. So in primates, uric acid is the excreted end product of purine catabolism in primates, birds, and some other animals. So uric acid, so if we shift over here now, you follow this, my laser pointer over here, you can see that by primates, birds, reptiles, and insects, they end up excreting um, uric acid. A healthy adult human excretes uric acid at a rate of about 0.6 grams per 24 hours. The exerted product arises in part from ingested purines and in part from turnover of the purine nucleotides of nucleic acids. However, in most mammals and many other vertebrates, so you can see right here, most mammals, marine vertebrates, bony fish, um, uric acid is further degraded to allantoin. Right here, you can see allantoin right there. by the action of urate oxidase right there. In other organisms, the pathway is further extended, and you can actually see it right here. So, bony fish um, would use the enzyme allantoinase right here to get allantoinate. And then you can also have uh, allantoicase to end up with urea. And then you can have urease to just get your final NH4 plus product that marine invertebrates end up excreting. It is worth mentioning, though, that primates excrete much more nitrogen, actually, as urea rather than uric acid, as you can see over here. And this is mainly because um, via the urea cycle. Um, So, shifting gears a little bit over here, we're going to look at the degradation of pyrimidines. So, the pathways for degradation um, of pyrimidines generally lead to NH4 plus, whereas before we were leading to uric acid for purines, um, and we were eventually getting to allantoin for most mammals. Bony fish, we're going to get allantoitate. And then amphibians and fishes, we're going to get urea. And then marine invertebrates, we're going to get NH4 plus. From again purines for pyrimidines, like I said, degradation generally leads to NH4 plus. So the production, um, and then eventually from there, urea synthesis from here. So a good example is actually thymine. Thymine, for example, is degraded to the final molecule down here. So methyl malonyl semialdehyde, um, an intermediate of a valine catabolism, and it is further degraded through propionyl CoA and methyl malonyl CoA to eventually end up to get succinyl CoA. Um, so something that we can actually talk about, try to relate to some diseases, is actually this degradation of pyrimidines. So we have these genetic aberrations in human purine metabolisms, which have been found, and some of them come with some serious consequences. So, for example, a classic example is adenosine deaminase deficiency, or ADA deficiency, which leads to severe immunodeficiency disease, um, in which T lymphocytes and B lymphocytes do not develop properly. Um, so this lack of ADA leads to a huge increase, approximately about 100-fold increase in the cellular concentration of DATP. And if we remember correctly, last time we said that a high concentration of DATP leads to our enzyme being unfolded correctly. So it ends up being a strong inhibitor of our enzyme ribonucleotide reductase, right? Because if we remember, we have high concentrations of DATP. We get that unfolding. We go from that alpha 2 beta 2 complex to that alpha 4 beta 4 ring like structure with alternating alpha and beta subunits. And when those subunits are alternating in a ring structure, we have exposure of these different or varying um, um, sections that are now exposed to solvent or aqueous environment, right? Where traditionally those hydrophobic areas would not be. So therefore, the structure now leads to a mishap in the function, and the only way to get it back, like we learned a little bit before, is to have low levels of DATP in our system. Our ring structure would then dissolve. It would go back in that reversible direction. We end up with that wonderful alpha 2 beta 2 um, unit that we know so well, and it can proceed and do what it needs to do. So what ends up happening is, with these high levels of DATP, we produce a general deficiency of other DNTPs. So all other DNTPs end up decreasing in T lymphocytes. Ooh, excuse me. So the basis for B lymphocyte toxicity is less clear. Individuals with ADA deficiency lack an effective immune system and do not survive unless isolated in a sterile, quote unquote, like, bubble environment. So these people have to be can't be exposed to the common cold, can't be exposed to the flu, bronchitis. They can't get an infection, if they get their fingers cut, and some grimy bacteria gets in there, or they have an open cut and they go in the water and swimming. They have absolutely no um, immune system, so they essentially have to live in this, sterile bubble environment. Um, so ADA deficiency was one of the first targets in human gene therapy back in the 90s. I think it was something like 1990 when they started looking at these therapy trials, which ended up yielding mixed results. But more recent trials, some ADA deficient individuals have regained a normal immune function after being given a functional gene for ADA, right? So it's just gene therapy, right? So if you have a functional ADA, functional gene for ADA, hopefully this individual can build an immune system. So shifting again, continuing forward, we're going to look at some purine and pyrimidines bases, which are recycled by salvage pathways. So free purines and pyrimidine bases are constantly released in cells during metabolic degradation of nucleotides. So what we end up having is we end up having free purines are in large part salvaged and reused to make nucleotides in a pathway very simpler or much more simpler than the de novo synthesis of purine nucleotides that we went through a little bit earlier in the previous lecture. And one of the primary salvage pathways consists of a single reaction catalyzed by adenosine uh, phosphorylribosyltransferase transferase. Oh my goodness, another difficult one to say. In which free adenine, so you can see right here, free adenine reacts with PRPP to yield the corresponding adenine nucleotide, which ends up being AMP plus. You get PPI as a byproduct from that. And another example of this is free guanine and hypoxanthine. So right here we can see from the hypoxanthine. So hypoxanthine is the deamination product of adenine, are salvaged in the same way by um, hypoxyanthine guanine phosphoribosyl transferase. So this is the enzyme that ends up salvaging free guanine and hypoxyxanthine. So a similar salvage pathway exists for pyrimidine bases in microorganisms and possibly in mammals as well. So this is one of those times that we can actually look at some medical, I guess, related things, and we can actually put together the things we've been learning and look at some, medically related information. So there is a genetic lack of hypoxyxanthine guanine phosphoribosyl transferase activity, which is seen almost exclusively in male children, and it ends up resulting in a very bizarre set of symptoms called Lesch syndrome. Um, I should have put it on the slide. I'll try to add it after. Lesch I'll try to type it out actually. Um, oh, this was not a smart idea. Actually, I'll just type it out later. So this Lesch syndrome, children with this genetic disorder, um, and typically this disorder, manifests itself by the age of, 2, maybe 3 years old. These children end up being poorly coordinated and have severe intellectual defects. And in addition to this, these children, like I said, male children, um, end up being extremely hostile, um, and end up showing some sort of like compulsive, self-destructive tendencies. Um, so they end up harming themselves. They end up, biting off their fingers. Some children have been shown to, bite off their toes, their lips. So they have this severe um, manifestation or compulsive disorder to self harm. And it has to do with um, this pyrimidines. So this sad and unfortunate and devastating effects of Laskanayan syndrome illustrates the importance of salvage pathways. So hypoxyxanthine and guanine arise constantly from the breakdown of nucleic acids, and in the absence of hypoxyxanthine, guanine, phosphorybosyl transferase, PRPP levels rise and purines are overproduced by the de novo pathway, which ends up resulting in high levels of uric acid production and gout like damage to tissues. So, what ends up happening is the brain is especially dependent on these salvage pathways, and this may account for the central nervous system damage in the children with Leschanayan syndrome. Um, and unfortunately, as far as I know, there's no real treatment for this right now. But the syndrome, would be a great candidate, right, for potential therapy, for gene therapy, right? If we could just, replace the genes that are malfunctioning and not making these enzymes properly, so that we can actually, um, you know, use these salvage pathways and help these individuals, these young children regain a sense of normality in their lives. So, continuing on this like biomedical application of nucleotides, nucleosides, we're gonna actually shift our focus now to gout and see how this actually works. So, gout is a disease of the joints caused by an elevated concentration of uric acid in the blood and tissue. So, excess uric acid ends up causing gout. Um, so what ends up happening is that you end up having your joints become inflamed, they become very painful, and they end up becoming like arthritic, like right, that inflammation of your joints, which causes arthritis. It's hard to move, um, and this is due or owing to the abnormal deposit of sodium urate crystals. So you end up having these sodium urate crystals being deposited into these joint like areas, which ends up causing them to be inflamed, painful, and this arthritis like um, um, pain. Um, Another part of the body that is affected as well ends up being the kidneys. So the kidneys are also effective as excess uric acid is deposited in kidney tubules. And gout occurs predominantly in males. It can happen in women, but again, mostly seen in males. And its precise cause is not known, but it often involves an under excretion of urate, um, a genetic deficiency of one or another enzyme of purine metabolism may also be a factor in some cases, and that's why we're going to highlight it right now. So gout is effectively treated by a combination of nutritional and drug therapies. Um, Lots of foods especially rich in nucleotides and nucleic acids, such as liver or glandular products, are withheld from the diet. Um, Major alleviation of the symptoms is provided by this drug called um, allopurinol. As you can see right here, you can see my pointer highlighting over. So what allopurinol does, it inhibits xanthine oxidase. So it inhibits xanthine oxidase, um, the enzyme that catalyzes the conversion of purines to uric acid, like we learned it before. So you can see right here, allopurinol inhibits xanthine oxidase and ends up forming this molecule over here called oxypurinol. Um, So what ends up happening is that this allopurinol is a substrate of xanthine oxidase, which converts allopurinol to this oxypurinol, or you can also call it like an alloxanthine. Um, so this oxypurinol inactivates the reduced form of the enzyme by remaining tightly bound in its active site. So when xanthine oxidase is inhibited, the excreted products of purine metabolism are xanthine and hypoxyxanthine, um, which are way more water soluble than uric acid and less likely to form these crystalline deposits. So from here, allopurinol was developed by a famous scientist, Gertrude Elion and George Hitchinsons. Um, which they also developed this famous medicine, what ended up used in treating people with genital or oral herpes infections and other purine analogs, which were also used in cancer therapy. So if we look at this, figure a little bit better, allopurinol is an inhibitor of xanthine oxidase. So this hypoxyxanthine is the normal substrate of xanthine oxidase. So you can see over here, this hypoxyxanthine is the normal substrate of this enzyme, xanthine oxidase. Um, Only a slight alteration in the structure of hypoxyxanthine can actually be seen. Um, you can see that shift right here, right? You can see you have the CH to this N group right here, and you end up forming this N to the CH group right there. So this yields the medically effective enzyme inhibitor allopurinol. And at the active site, allopurinol is converted to oxypurinol, a strong competitive inhibitor that remains tightly bound to the reduced form of the enzyme. And again, less likely to form crystalline deposits, and gives these individuals that had gout and this painful lifestyle before the chance once again to enjoy um, a somewhat regular and normal life, with pain free, which is the wonders of modern medicine. So um, this medication was made in the early 1900s. So good. All right. Well, continuing on with this whole, I guess, biomedical application of this nucleotide pathway, we're going to focus on cancer targets now. So many chemotherapeutic agents target enzymes in the nucleotide biosynthetic pathways, right? So we can kind of start off with a broad definition where the growth of cancer cells isn't controlled in the same way as cell growth in many normal tissues, right? Cancer cells end up having a greater requirement for um, many macromolecules, right? Think about carbohydrates, right? They're fueling their cells with glucose all the time. That famous Warburg effect, where cancer cells shift their energy source to um, not oxidative phosphorylation. You know, but this is where I kind of just interject and say that not all cancer cells have the same requirements, right? Some of them rely on oxidative phosphorylation for their energy requirements, while the other ones may not at all, may not use it at all. And then there's many cancer cells in between that use varying degrees of each, of um, each pathway to sustain themselves. One of the other things is that people sometimes overlook is lipid composition in these cancerous cells. Uh, Cancerous cells are rapidly dividing and rapidly growing, proliferating, so they require rapid access or easy access to um, lipids. Sometimes immature lipids are used. So you have, a different lipidomic composition of healthy cells versus cancerous cells. And sometimes, like we learned in the previous units, um, the composition of lipids provides these functional platforms for proteins to efficiently work, especially proteins that are associated with the membrane. So there's a lot of things that go on with cancerous cells, right? And one of them is that cancer cells have a greater requirement for nucleotides as precursors. of DNA and RNA, and consequently, they're generally more sensitive than normal cells to inhibitors of nucleotide biosynthesis. So, um, you know, there's many growing, chemotherapeutic agents that are growing nowadays that are important for cancer and other diseases. They act by inhibiting one or more enzymes in these specific pathways. Um, and we're going to talk about them here a little bit. So this first set that we're going to talk about here of agents, these are compounds that inhibit glutamine aminotransferases. Um, and if you guys remember that glutamine is a nitrogen donor in at least half a dozen separate reactions in nucleotide biosynthesis. So the binding sites for glutamine and the mechanism by which NH4 plus is extracted are quite similar in many of these enzymes. So most are strongly inhibited by glutamine analogs such as azacitidine over here and um, acivacin. Oh God, can't say that one either. So azacitidine over here was first characterized by John Bushman in the 50s or 60s, and was one of the first examples of a mechanism-based enzyme inactivator. So acivin shows uh, promise as a cancer therapeutic agent, and it's constantly used. So it's just to summarize, these analogs of glutamine interfere in several amino acid and nucleotide biosynthetic pathways. So, you know, if these cells are proliferating uncontrollably, you want to slow down the building blocks that are actually able to um, make these cells, right? So DNA, RNA, lipids could be one of them, right? There's many targets you can use, but specifically here, we're just looking at these nucleotide um, inhibitors. So another useful cancer target that we have here for pharmaceutical agents are thymidylate synthase. So you can see right here, this is thymidylate synthase. Um, and dihydrofolate reductase. So this enzyme over here. These are enzymes that provide the only cellular pathway for thymine synthesis. And this right here, and we'll go through in a little bit how thymine synthesis actually works. But one inhibitor that we want to focus on over here um, is this molecule over here called fluorouracil. So fluorouracil is an inhibitor that acts on thymidylate synthase, um, and it's an important chemotherapeutic agent. Uh, so fluorouracil itself is not the enzyme inhibitor. Um, in the cell, salvage pathways converted to the deoxynucleoside monophosphate F dump. And we'll go through that pathway in the next slides, which binds to and inactivates the enzyme. So if we just kind of focus on this graph now just a little bit, we can see that during thymidylate synthesis, we have this N5, N10 um, methylene tetrahydrofolate is converted to 7,8 hydrofolate. So the N5, N10 methylene tetrahydrofolate is regenerated in two steps. And this cycle is a major target for several chemotherapeutic agents. But if we look over here down at this molecule over here of um, fluorouracil and uh, metho- methotrexate. They're important chemotherapeutic agents. So in cells, fluorouracil is converted to F dump, which inhibits the thymidylate synthase over here. And then you have methotrexate, a structural analog of tetrahydrofolate, inhibits dihydrofolate reductase, this one over here. Um, and the shaded amino in methyl groups replace a carbonyl oxygen and a proton, respectively, in folate. Um, And then over here we have tri trimetho prim trimethoprim, which is just a type binding inhibitor of bacterial dihydrofolate reductase. And this just was just developed as an antibiotic, and it just kind of has a familiar structure as the other ones. So they were just grouped in together with this example. So, like I said, we want to talk about a little bit more about fluorouracil and how it actually works. So, in the cell, salvage pathways um, convert it to deoxynucleoside monophosphate, which is this F dump over here, which binds to and inactivates the enzyme. And inhibition by F dump is a classic example of like a mechanism-based enzyme inactivation. And another prominent chemotherapeutic agent is methoxytrate that we talked before. It's an inhibitor of dihydrofolate reductase. Um, so this folate analog acts as a competitive inhibitor. The enzyme binds methoxytrate with about 100 times higher affinity than the hydrofolate. Um, and another example of another chemotherapeutic agent is aminopetrin, and it's a related compound that acts quite similarly. Um, and before I continue on with this pathway here, I just want to kind of quickly go over um, the drug I kind of just skipped over kind of fast, trimethoprim. So the the medical, I guess, potential of inhibitors of these nucleotide biosynthesis, it's not really limited to, cancer treatment only. So all fast-growing cells. So whether it's bacteria, protists, they can all be potential targets. So trimethoprim, which, like I said earlier, is an antibiotic developed, a long time ago, binds to bacterial dihydrofolate reductase nearly 100 times better than to the mammalian enzyme. And it's used to treat, certain urinary and middle ear bacterial infections, such as we have, parasitic protists, um, such as, diseases that cause African sleeping sickness. Um, so these diseases, such as, African sleeping sickness, they lack pathways for de novo nucleotide biosynthesis and are particularly sensitive to agents that interfere with their scavenging of nucleotides from the surrounding environments using salvage pathways. So several similar, analogs have been shown and been promising for the treatment of African sleeping sickness and related diseases. Um, and this has all been, possible because of the advancements we've had in understanding metabolism and enzyme mechanisms. Okay, so let's go through this pathway right now and kind of just go over it. So we have the conversion of dump, so DUMP, to DTMP, and its inhibition by FDUMP or FDump. So the left-hand side over here, you can see my cursor, is the normal reaction mechanism of thymidylate synthase. So the nucleophilic sulfhydryl group, um, right over here, there we go, we're zooming in now, um, contributed by the enzyme um, in reaction 1, and the ring atoms of dump, so DUMP, take part in the reaction that's shown in the red that's happening right there. Um, and you can see over here in this little one where my laser pointer is, you have the B and your two dots right there, denotes an amino acid side chain that kind of acts as a base to abstract a proton after step 3, and we'll see that a little bit later. So in step 1, what you have is the enzyme thiolate adds at C6 of dump uh, a Michael type addition, so N10 is protonated, and the N5 inium ion is formed uh, from methylene H4 folate, as you can just see from this little text box right here. And then in step 2, you have that C5 carbanion adds to the N5 iminium ion. And then step 3, we have that methylidine is formed at C5 of the pyrimidine and N5 is eliminated to form the H4 folate. So what happens here is the hydrogens derived from the methylene group of the N5, N10, um, methylene tetrahydrofolate, which are shaded in gray, as you can see over here, shaded in gray. And then what happens now in step 4, which is the last step, you have this 1,3 hydride shift, and it moves a hydride ion shaded in this, this light red color over here, um, right here. Um, from C6 of H4 folate to the methyl group of thymidine, resulting in the oxidation of tetrahydrofolate to dihydrofolate. And this hydride shift is blocked when F dump is the substrate on the right hand side, which we'll see right now. So what happens is you have step 1 and step 2 proceed normally, but result in a stable complex consisting of F dump linked covalently to the enzyme and to tetrahydrofolate that inactivates this enzyme. So as you can see over here, on the DUMP side, this hydrogen group right here is regular and is a hydrogen, and on the F dump side, it's actually a fluorine group right here. So when you hit the last step right here, step 2, and about to begin step 3, you have a dead end covalent complex. So it results in this stable complex, which is then, like I said earlier, covalently linked to the enzyme and to tetrahydrofolate that ends up inactivating this enzyme, and you can't move forward. So you can't actually do, step 3, and you can't do step 4 to get your DTMPs, which is your final molecule. And this is kind of how these anti-cancer um, mechanisms tend to work. So in the previous slide, we were talking about these cancer targets, right? And I was mentioning thymidylite quite a bit. So in this slide, we're just going to go over what it is, and we're just going to talk about how thymidylite is derived from DCDP and DUMP. So DNA, it contains thymine rather than uracil, as we know. Um, and the de novo pathway to thymine involves only deoxyribonucleotides. So the immediate precursor of thymidylite, or DTMP, is DUMP, or dump. In bacteria, um, this pathway to dump, or DUMP, begins with the formation of DUTP. So either by some deamination of DCTP, or you have the phosphorylation of DUTP. Um, UDP, sorry. So we can see that kind of down here. So this is the biosynthesis of thymidylite over here. So the pathways are shown, um, beginning with the reaction catalyzed by ribonucleotide reductase. So you can see right here. So we start off with CDP or UDP. We have ribonucleotide reductase gets us to DCDP or DUDP. From there, we have nucleoside diphosphate kinase gets us to DCTP and DUTP. And then we have a deaminase reaction that goes from DCP to DUTP. And then from there, it just moves forward. So we have DUTPase to dump, and DUMP, we have our enzyme thymidylate synthase to get um, M, DTMP. So the DUTP is converted to dump by DUTPase, as we went over that already. And this reaction must be actually quite efficient, but efficient in a different way. It must be efficient in the sense that it must keep these DUTP levels right here quite low, actually, to prevent incorporation of uridylate um, into DNA, right? You don't want uracil going into DNA, right? Because we have thymine in DNA. So we got to keep these pools quite low. So the conversion of DUMP right here to DTMP is catalyzed by the enzyme thymidylate synthase, which was the enzyme that the cancer therapeutics in the previous slides were actually targeting. And we'll kind of go through the pathway more over here in a little bit. So this is catalyzed by thymidylate synthase, like I said. So a one carbon unit at the hydroxymethyl. So over here, um, so over here, a one carbon unit at the hydroxymethyl oxidation level is transferred from this N5N10 methylene tetrahydrofolate to DUMP molecule over here. Um, then it's reduced eventually to a methyl group. So the reduction occurs at the expense of oxidation of tetrahydrofolate um, to dihydrofolate, right? So we go from this methylene tetrahydrofolate and we go to this reaction that is this dihydrofolate over here, this compound, sorry, which is unusual in tetrahydrofolate requiring reactions. Um, so the hydrofolate is reduced to tetrahydrofolate by dihydrofolate reductase. So if we want to go back, we can just use this dihydrofolate reductase over here to get tetrahydrofolate back. Regeneration that is essential for the many processes that require tetrahydrofolate, specifically in plants, and at least one protist that is kind of known. And then we have thymidylate synthase and dihydrofolate reductase, which reside on a single bifunctional protein. So again, that reoccurring theme that um, different active sites and different polypeptides all play together on these multi-complex enzymes that all serve to be efficient to convert different molecules and different reactions as quickly and as efficiently as possible. So if we look into this mechanism just a little bit into more detail, we see that we have serine hydroxymethyltransferase is required for the regeneration of this N5N10 methylene form of this tetrahydrofolate right here. So we need the input of a serine amino acid. We need it stabilized by a PLP, which is usually just used to stabilize the molecule. We have a leaving group of glycine that comes in, and it's the serine that actually provides this CH2 molecule here. And just something that we should note over here is that in the synthesis of DTMP, so our molecule over here, all three hydrogens of the added methyl group are derived from the N5, N10 methylene tetrahydrofolate. So you can see it right here, right? This shaded gray and this shaded orangey red color end up over here on the DTMP. So everything um, comes from this N5, N10 methylene tetrahydrofolate. So our CH2 comes from our serine, and this hydrogen over here comes from when dihydrofolate reductase uses NADPH to get NAD plus. And to throw in some of these, more biomedical type of, examples, we kind of want to talk about how about 10% of the population, of human population, and up to even 50% of populations in third world or impoverished communities suffer from a folic acid deficiency. So, when a deficiency is severe, the symptoms can include things such as heart disease, cancer, and even some sorts of brain dysfunction. And at least some of these symptoms can arise from a reduction of thymidylate synthesis, leading to an abnormal incorporation of uracil into DNA. So when uracil is recognized by DNA repair pathways, it's actually cleaved from the DNA, and the presence of high levels of uracil in DNA leads to strand breaks that can greatly affect the function and regulation of nuclear DNA, which ultimately causes the observed effects of the heart and the brain, as well as increase mutagenesis that lead to cancers later on in life for these populations. So, kind of to go over a summary of some of the concepts that we should have learned from the previous two lectures. We'll kind of just go over them right now. So, the purine ring system. So, we should have learned that the purine ring system is built up step by step, beginning with five phospho ribosylamine. The amino acids, glutamine, glycine, and aspartate, furnish all the nitrogen atoms of purines, and two ring closure steps form purine nucleus. Um, Something else we should have learned is that pyrimidines are synthesized from carboxyl phosphate and aspartate together, and ribose 5 phosphate is then attached to yield the pyrimidine ribonucleotides, right? So one is built step by step, the other one is an attachment later on. We should have also focused on nucleoside monophosphates, which are converted to their triphosphates by enzymatic phosphorylation reactions. These ribonucleotides, they're converted to deoxyribonucleotides by ribonucleotide reductase, an enzyme which has, regulatory characteristics and novel mechanisms, right? The radicals that we talked about from the tyrosine 122. Um, we also should have looked at the thymine nucleotides are derived from DCDP and DUMP. Some of them examples that we saw were in cancer related as well. We should have also looked at that uric acid and urea are the end products of purine and pyrimidine degradation. Free purines can be salvaged and rebuilt into nucleotides. And because of that, we can actually have some genetic deficiencies in certain salvage enzymes that end up causing serious disorders such as ADA and Lesch-Nyhan syndrome, which is right here. We also kind of quickly talked about the accumulation of uric acid crystals in the joints, possibly causing another genetic deficiency such as gout, which ends up resulting in gout. And lastly, um, enzymes of the nucleotide biosynthetic pathways are targets for an array of chemotherapeutic agents used to treat cancer and other diseases, such as the antibiotics that we quickly went over. And that kind of sums up um, biosynthesis and degradation of nucleotides. And like I said at the beginning of the lecture, the next topic will be biosynthesis of carbohydrates in plants, because it just makes the most logical sense to go that way, and then go into lipids. And then from lipids is actually a quick transition into some of the special topics we can get into really quickly. We can go into diseases right after that because of the way lipid biosynthesis actually happens or occurs in the body. And with that, have yourself a good rest of your week. Have a good weekend, and I will see you guys bright and early Monday morning, 8:30 in person at a regular scheduled time, and we shall continue lectures. Um, until then, bye and take care.